Welcome to The Explainer. Today, we're unpacking a topic that touches nearly all of our lives, yet, honestly, is often deeply misunderstood. We tend to think of cancer as a random, chaotic event, right? It's kind of like a lightning strike. But when you really look at it through the lens of biochemistry, it's anything but random. It is a fascinating, highly structured, multi-stage biochemical corruption of our very own cellular programming. A healthy cell doesn't just suddenly become a threat out of nowhere, it gets reprogrammed. It essentially goes rogue. And today, we're going to dive into exactly how that happens.
So, here is our roadmap for today. We'll be moving through 6 main areas. First, defining tumors and cancer. Second, the 3 stages of cancer. Third, triggers like carcinogens and causes. Fourth, the molecular battleground. Fifth, how cancer actually fuels itself. And finally, number 6, the medical counterattack.
All right, Section 1, defining tumors and cancer. Let's get into it.
Before we can really understand the medical war being waged, we have to understand the enemy. Over in pathology, the strict academic term for this rogue growth is a neoplasm, which literally just translates to new growth. But the absolute key takeaway here is that this uncoordinated growth persists even after the initial trigger is completely gone. So, a neoplasm isn't just an injury or some temporary reaction, it's a profound genetic alteration. The cell's foundational instruction manual has been permanently rewritten.
Now, in everyday language, a neoplasm is usually just called a tumor, but we all know not all tumors are created equal. The crucial thing is understanding the difference between a benign tumor and a malignant one. Benign tumors are your slow-growing, highly localized masses. They stay put, and biochemically, their cells actually look pretty normal. Their cell cycle controls are largely preserved. Malignant tumors, on the other hand, which is what we actually call cancer, are the true rogue agents. They divide incredibly rapidly, their cells are highly abnormal, and most dangerously, they're invasive and metastatic. They absolutely refuse to be contained.
At the tissue level, the physical difference is pretty stark. A benign tumor stays neatly zipped up inside a fibrous capsule. It might push against things, sure, but it never infiltrates. A malignant tumor has zero restraints. It actively breaks through normal biological boundaries. It can ulcerate the surface, and terrifyingly, it invades local lymphatic vessels and bloodstreams. That right there is exactly how it manages to metastasize to the rest of the body.
Moving on to Section 2, the 3 stages of cancer. So how does a perfectly healthy cell transform into this invasive mass? Well, it's a long-term process that always follows 3 distinct stages. First up is initiation. This is a permanent, irreversible mutation in the cell's DNA. The blueprint gets damaged, but at this point, the cell still looks normal. Second is promotion. This is where things really start to escalate. Promoting agents, things like hormones or chronic inflammation, stimulate that initiated cell to divide rapidly. This stage is, well, it's actually reversible, but it manages to create a massive clone army of mutated cells. Finally, we hit progression. This is the absolute point of no return, where these cells acquire truly malignant properties, like the ability to invade surrounding tissues and completely resist natural cell death.
It's almost like a cascading avalanche. Starting from a single normal cell line, an agent comes in and damages the DNA. That's our initiation phase. Then as promoters force that one damaged cell to multiply, we see this massive clonal expansion. It essentially creates an expanding family tree of mutated cells. Finally, in progression, they clump together into a malignant tumor, accumulating more and more genetic errors until they become this aggressive, unstoppable force.
Section 3. Triggers, carcinogens, and causes.
So, what actually pulls the trigger on that very first DNA mutation? You might assume it's mostly just bad underlying genetics, but the staggering reality is that environmental factors are by far our biggest threat. Chemical carcinogens alone are responsible for a massive 80% of cancers. We're talking about substances found in tobacco smoke, industrial pollutants, and even certain food preservatives. Beyond chemicals, we've got radiant energy, like UV rays from the sun literally tearing at our DNA, as well as oncogenic viruses like HPV, and even prolonged hormonal imbalances. Any of these can force that initial, irreversible DNA error.
There's a deeply tragic biological irony when it comes to chemical carcinogens. Many of the chemicals we ingest aren't actually dangerous at first. They're basically inert. But when they enter our cells, our liver's own detoxification enzymes step in to try and metabolize them. In doing so, our own bodies accidentally activate these chemicals into highly reactive molecules called electrophiles. And those electrophiles, they aggressively attack our DNA. We essentially armed the bomb ourselves.
Section 4. The molecular battleground.
The resulting tumor is really the fallout of a microscopic war taking place deep inside the nucleus. Think of cellular division as the ultimate tug-of-war between two main categories of genes. On one side, we have normal growth signals, which, when they mutate, become what we call oncogenes. These cause a gain of function, meaning they constantly scream at the cell to divide, divide, divide. On the other side of the rope, we have tumor suppressor genes. These guys are the peacekeepers. They normally regulate the cell cycle and hit the brakes on growth, but in cancer, they suffer a loss of function. The peacekeepers are effectively taken out.
To really cement this concept, imagine a normal cell is a car. An active oncogene is a car with a brick stuck on the gas pedal. It is constantly accelerating. Normally, you have brakes, your tumor suppressor genes, which can stop the car anyway, despite the stuck pedal. But if you have an active oncogene and you simultaneously lose your tumor suppressor genes, well, you now have a car with the pedal to the metal and absolutely no brakes. A high-speed catastrophic crash is literally inevitable.
If you only remember one single molecule from today's explainer, make sure it's the p53 gene. It is famously known by scientists as the guardian of the genome. It is the single most vital tumor suppressor gene we have in our bodies. When p53 detects DNA damage, it actively halts the cell cycle just to allow for repairs. If the damage is too severe to fix, p53 forces the cell to gracefully commit suicide, a process called apoptosis, in order to protect the rest of the body. So when a carcinogen mutates and destroys p53, the cell loses its ultimate guardian and the road to cancer is wide open.
Section 5. How cancer fuels itself.
Okay, so with the brakes cut and the gas pedal totally floored, this rogue colony faces a massive logistical nightmare. It needs a ridiculous amount of fuel to survive and build out its clone army. To solve this energy crisis, cancer cells drastically alter their metabolism in a weird phenomenon known as the Warburg effect. Normally, human cells use oxygen to very efficiently extract energy from glucose, but cancer cells are greedy and they're in a massive rush. Even when there is plenty of oxygen available, they switch over to a less efficient but much faster process called aerobic glycolysis. They start consuming staggering amounts of glucose, prioritizing rapid ATP energy production, and, crucially, generating the biological building blocks, like extra lipids and nucleotides, that they desperately need to physically construct new cells at breakneck speed.
Normally, a cell carefully and methodically processes glucose through the mitochondria for a massive payout of 38 ATP. It's a highly efficient system, but a cancer cell? It bypasses this almost completely, yielding a terrible 2 ATP per glucose. Why on earth would it do that? Because by churning through glucose at a massive, greedy rate, it rapidly generates lactate, which gets incorporated right into new biomass. It completely sacrifices efficiency for pure, unadulterated speed and building materials, supporting rapid, unchecked cell proliferation.
And finally, Section 6, the medical counterattack. So how does modern medicine actually strike back against such a resilient, hijacked system? We attack its fundamental biochemistry using 4 main pillars. First, we physically cut the localized mass out with surgery. Second, we burn its DNA with radiotherapy. Third, we use chemotherapy to strictly poison the cell replication cycle, using drugs that mimic natural metabolites or alkylate the DNA to physically block division. And finally, the fourth pillar, we use biological therapies. These are things like specific enzymes that starve the tumor of certain amino acids or drugs that block the very hormones promoting its growth.
What's really fascinating is that these medical treatments essentially use the cancer's own vulnerabilities against it. Take radiotherapy, for example. High-energy particles are deliberately fired at the cell to cause massive, non-repairable DNA damage. Now, because the cancer cell has likely lost its p53 repair mechanisms in its rush to grow so fast, it just cannot fix this sudden onslaught. Its own broken repair system causes a catastrophic cellular failure. We are literally using its biological recklessness as a weapon to destroy it.
But of course, the war is far from over. Cancer cells are incredibly unstable by nature, meaning they continuously mutate, and that allows them to develop resistance to our chemotherapies and radiation over time. So I want to leave you with this final provocative thought. Understanding this deep biochemistry, the oncogenes, the Warburg effect, the P450 enzymes, is really just our opening move. As these rogue cells continue to evolve to evade our best medical counterattacks, how will the next generation of targeted biochemical therapies manage to stay one step ahead in this ultimate microscopic arms race? Thanks for watching this explainer, and keep questioning the science around you.